Do you prefer, Allegra, if I leave you this view of the group so you can see some faces of how they react? Yeah? Okay, that
Yeah,
sounds good.
that's good. Yeah.
Okay, let's welcome back, Allegra, please. You. Hello, everybody. Okay, Allegra, the stage is against yours. Welcome back.
Thank you. All right. So can you all hear me again? Okay. I will take that as a yes. Um, all right. So yeah, thank you again for the reintroduction. So now that I've told you a bit about the basics of mass spectrometry and data acquisition, hopefully we can go a bit deeper into really my research interests. Okay. And so yeah, I'm quite excited today for the research talk, maybe more excited for the research talk than the didactic talk, in all honesty. And so today I'm going to be telling you about our quest to find metal binding molecules. And I'll be telling you about a strategy that we call native electrospray metabolomics that has enabled us to start doing this. And then I'll sort of give you my vision for what I think is next, right? I'm really sort of hoping to find many metal binding molecules, because I think this is sort of an underappreciated class of molecules that plays really important roles across biology and especially in microbial systems. Okay, but I like to start my talk with this picture. Firstly, because I'm coming to you from Denver, Colorado, where we have lots of beautiful nature, lots of rocks for climbing, but also because I'm a chemist, right? And so if I look at a beautiful scene like this through that lens, I realize that actually each of the environments we can think of as just sort of mixtures of molecules working together in organized fashions to support each environment, including the human life, right? Now, typically when people think of biological chemistry, they think of organic elements like carbon, nitrogen, oxygen, and sulfur. However, I am sort of at heart a bioinorganic chemist. And so when I look at a living system, I think not only of those organic elements, but I also think about the metals, right? And why do I think about the metals? Well, metals are actually also essential for life. Metals, as many of you probably know, play essential roles in respiration and electron transfer. They play critical roles in oxygen transport, right, in heme, and they are essential in DNA replication and repair. Additionally, in plants, metals play key roles in photosynthesis. Okay, laser pointer ready. And in other organisms, they're essential for nitrogen fixation. Okay, now though, if we sort of change our perspective and think perhaps like a microbiologist, we realize that also in each of the environments pictured here, there exist many, many microbes, right? So for example, on the leaves of these trees, there are whole microbial communities, right, working together as an ecosystem, and similarly in the gut of this man. And so I've told you metals are essential for life, right? We can obtain metals through our diet, right? Many of you know that we get metals from red meat or leafy green vegetables, right? But the question then becomes, how can microbes actually acquire metals, right? Because, well, they can't just eat a steak, for example. And so actually microbes have really cool and intricate mechanisms for acquiring metals, especially in environments that are metal deficient. And so one of the ways that microbes can actually acquire metals is through the biosynthesis and then the secretion of these secondary metabolites, natural products that we know of as siderophores or metallophores. Okay, now siderophores are sort of complex molecules. They bind iron 3 with very high affinity and very high specificity often. Metallophores bind non-iron metals, right? And so there's been recent discovery of a number of new chalcophores which bind copper, zincophores which bind zinc, etc. And I'll actually tell you some of those stories in a minute here. But what microbes can do is they can secrete these molecules into their external environment. And because these molecules bind metals with very high affinity and specificity, they can bind them even when the metal levels are very low. And then these molecules can be recognized by a receptor, taken up by a transporter, and then they can actually be delivered to the microbe, right? And so the molecule and the metal will dissociate, and the microbe can then acquire metal. Now, I think the chemistry of this is just fascinating, but in addition to being simply very cool, these metal binding molecules actually have a number of roles across biology and a variety of applications, right? And so these metal binding molecules have actually been found recently to mediate interactions between microbes. So for example, in the human gut, microbes can produce a siderophore, and then actually other microbes that do not produce that molecule can utilize the molecule, right? So it's a way that microbes can actually share molecules with one another. Additionally, these molecules can mediate host microbe interactions. So for example, bacterial siderophores can actually interact with host proteins, and this can sort of provide signaling as to iron status. For example, these molecules are also important in mediating interactions within ocean ecosystems and soil and leaf microbiomes. And we can also take advantage of these metal binding molecules for energy applications and even for bioremediation of contaminated environments. Okay, so these molecules have a lot of important roles and applications. And so when I started my postdoc a few years back now, actually Daniel and I, in collaboration, were really interested in understanding how do metal binding molecules shape interactions within host-associated microbiomes. So we wanted to explore that, but it quickly became apparent to us that at the time, high-throughput methods for finding metal interactions with small molecules were very limited. Now, why might this be the case? Well, we hypothesized that low pH and high levels of organic solvent used in chromatography, and specifically liquid chromatography, along with sample extraction, often destroy metal complexes. Additionally, when we perform extraction and chromatography, we're using low metal concentrations. And so this also disfavors metal complexation, pulling equilibrium away from the metal complex. And then finally, these molecules are often produced in small quantities, right? And so given then that we might even be looking for the metal adduct of a molecule produced in a very small quantity, this would be very close to the detection limit. So we might just not be seeing it in the context of a very complex sample. So how did we hypothesize actually addressing these issues? Well, we thought that we could actually alter our mass spec conditions. So now using native ESI, and we could take advantage of new computational strategies that were being developed in the Dorstein laboratory simultaneously at the time. And you'll actually hear some more about these computational strategies throughout this workshop. Okay, so we thought that we could tackle this problem using this two-pronged approach. And we were very excited to sort of come up with this little scheme. I don't know if there are any Metallica fans in the audience, but we call this Metabolomics in addition to native metabolomics. Okay, and so again, I just gave a talk on how we do MS data acquisition. And so your mind should be very fresh, but this is what our setup looks like, right? So we have our sample, we run liquid chromatography, followed by mass spectrometry, right? And so all the data that I'll be showing now was acquired on a Q-Exactive Orbitrap, right? And then we do a variety of data analysis. Okay, and so the native electrospray ionization strategy that we envisioned looked like this, okay? So we envision running our LC using standard conditions, right? So this would be a low pH. But then we thought that perhaps we could actually infuse in a buffer that would allow us to raise our pH to some controlled level. And the pH level that we would use would be the pH level that we hypothesize we'd observe maximal metal binding. Okay, so for example, a physiological pH of 7. At this point, once we raised up our pH to this level, we would actually infuse in our metal salt. Okay, now at this point, any metal metallophores or metal binding molecules would be poised to bind metal that was just infused. Okay, and then at this point, we'd actually send these complexes to the detector for analysis. Okay, now 2 things to note. So the first thing is that this metal binding step we hypothesized would not be 100% efficient. And so because of this, we hypothesized that we'd see both the proton adduct and the metal adduct. We thought we could actually take advantage of seeing both adducts in subsequent computational steps. The additional thing to make note of is the fact that we're doing this metal salt infusion after chromatography, right? And so we'll be seeing both proton and metal adduct at the same retention time. And this will also be very important in our downstream computational analysis. Okay, so in order to test this native metabolomics workflow, we first wanted to do this, or to try this on a simplified mixture of standards. Okay, and so here you can see various different siderophores, iron-binding molecules with different chemical structures. And so what we did is we prepared a mixture of these, and then we ran these samples using standard LC-MS, MS, low pH workflows, and then using our new native ESI workflow with our post-iron infusion. And so we were quite excited to see the data looking like this, right? So I've just highlighted one compound. This is ferrochrome. Now, in the absence of iron infusion, using standard LC-MS conditions, we see the presence of this proton adduct, and then we see our sodium adduct. We don't see any iron adduct. However, when we do our introduction of buffer, so we raise our pH, and then we do our post-LC iron infusion, what we see is the proton adduct in addition to a very prominent iron adduct, okay? And so that was quite exciting to us because, you know, even though this wasn't too exciting of a result, we know this is an iron binding compound. We are indeed seeing the iron adduct. It suggested that doing this post-LC iron infusion would allow us to see both proton and iron adduct, which meant that we could actually pair this native ESI workflow with the downstream computational analysis that we envisioned in order to find metal binding molecules, right? So this was a very simple proof of concept experiment. The next thing that we wanted to look at would be, could we actually find microbially produced metal binding compounds in samples that were more complex, or in cases where we actually had no idea what we were looking for, right? And so we thought that in order to do this, we could take advantage of molecular networking, specifically feature-based molecular networking in GNPS, and ion identity networking in MZMIND2. Okay, and so I think you'll hear much more about molecular networking in GNPS in the coming days. But just to kind of get you up to speed quickly, molecular networking can allow us to put molecules into context with one another, right? Because it actually reports on spectral similarity. Okay, and so let's look at this example now of how molecular networking actually works and what it is. So here again, we see that molecule that I keep showing, domoic acid, right? And so what we can do is we can actually fragment domoic acid here using HCD, right? So this is the MS2 spectrum of domoic acid. Okay, and so here we can see the left-hand side of the molecule is represented by this green peak. The right-hand portion is represented by this purple peak, and then the full molecule is is represented by this starred precursor ion. Now, if we look at the derivative of domoic acid that is modified here at this carboxylic acid into this ester, we see that the left-hand side of the molecule, has shifted by a mass of R, and the precursor, the intact molecule, has also shifted by a mass of R. And so these two MS2 spectra are quite similar, right? And so because of this, what we can do for purposes of molecular networking is we can actually represent each of these spectra as a circle with our precursor mass to charge ratio. We call this circle a node. And then what we can do is we can actually draw a line between these two circles. Now, this line reports on the fact that these two MS2 spectra are similar to one another, where we're actually measuring spectral similarity with a cosine score. Okay, now a cosine score of 1 means the two MS2 spectra are identical. A cosine score of 0 means they're completely dissimilar. And so the two nodes with a cosine score of 0 would not be connected, while the nodes with a high cosine score would be connected. And so what we can do next is we can build out a molecular network that reports on all of the connections between nodes, which tells us about spectral similarity and molecular similarity. Okay, then what we can do is we can actually pair that molecular networking with an additional step. And this would be where ion identity molecular networking comes in. So now what I showed you previously was looking at the MS2 level, right? We were only looking at those tandem MS-MS spectra. Now we want to look at the MS1 level, okay? And so what we can do is we can take our data set, we can do feature finding, which you'll also be talking about in this workshop. Then we can start to say, okay, here we have 4 features, right? What we can do now is we can look and do a retention time and a peak shape correlation between those features. So for example, we can see that features A and B occur at the same retention time, features C and D occur at the same retention time. Moreover, we can start to see that the peak shape of A and B are correlated, and the peak shape of C and D are also correlated. From there, then we can do a filtering step. So for example, if we're interested in looking at iron binding molecules, we'll be specifically looking for features that have retention time and peak shape correlations and also differ from one another by this mass delta, which is associated with the binding of iron 3. And so from there, we can draw in a different type of connection between nodes. So this connection will now be reporting on that sort of iron binding connection, right? So now this will essentially be not an MS2 similarity edge, but rather an edge associated with an MS1 mass delta, okay? So here I'm showing an orange dashed edge indicates likely binding of iron. So now we can look back at that mixture of standards, right, that I showed previously, and we can actually look at what the ion identity molecular network of that mixture looks like. And what you can see is for each of those molecules shown here, so for example, your cineobactin, we see that there is a very prominent iron binding event, okay? And so here I've actually colored this molecular network where purple refers to the proton adduct of each standard, orange refers to the iron adduct of each standard, other refers to just other features we observed in the MS. We're looking at our standard molecular networking edges, so our MS2 similarity edges as gray solid lines, and then the dashed lines are reporting on these MS1 mass delta edges, right? And so the orange dash means that we are observing iron binding. And so what you can now see is that these iron binding molecules are really jumping out of the page, right? We can really easily see them. And so this seemed quite promising for now taking this approach away from just looking at standards and actually using this approach on complex samples. Okay, and so the next 2 stories I'll tell you are just briefly about how we did that to make some interesting biological discoveries, and then I'll sort of tell you where we're going with this. Okay, and so I've chosen to first discuss one example that relates to human health, and then I'll actually go into describing an example that relates to the health of the open ocean. Okay, and so when we had this workflow sort of developed, we were quite excited because our collaborators at UCSD, so Huizi in the Manuela Raffatello lab at UCSD, made this interesting observation, right? And so he discovered that E. coli Nissl is able to outcompete the pathogen Salmonella in the presence of low levels of zinc. Okay, now this seemed quite curious because E. coli Nissl is actually a probiotic organism, right? Though mechanisms of how it actually is a probiotic are incompletely elucidated, right? And so we hypothesized that maybe E. coli Nissl was actually just better able to acquire metals under limitation than pathogens. And perhaps it's this ability that allows E. coli Nissl, the probiotic, to actually outcompete pathogens. Okay, and so we wanted to see if this was indeed the case, right? And so what Huey did is he actually created the genetic mutant that removed both the high affinity zinc transporter and the low affinity zinc permease in both E. coli Nissl and in Salmonella. And what he observed was that E. coli Nissl was still able to outgrow Salmonella in low zinc conditions, okay? And so from there, we hypothesized that E. coli Nissl was likely producing a small molecule that facilitated zinc uptake through this YBTX protein, okay? But now the question was, well, what is this small molecule? And is there a small molecule zinc binder produced by E. coli Nissl? And so that's where we thought that our method was actually the perfect method to answer this question. Okay, and so what we did is we took wild type and mutant. So with these transporters and this biosynthesis knockout, we took these organisms and we ran mass spec, right? And so what we did then is we ran them using our new workflow. Okay, sorry, technical difficulty. And so we ran wild type and knockouts in the presence of our native metabolomics method, right? So this would be using our modified pH and then doing a zinc infusion. Okay, and so here we're looking at a molecular family coming from the wild type E. coli Nissl. And so what you can see is, again, I've sort of colored these nodes and edges to help with visualization, okay? And so the dark blue nodes are proton-bound molecules that actually have some mass delta associated with the difference between zinc and the proton. These pink nodes are likely zinc-bound nodes, okay? And so what you can see is that looking at the wild type, it seems like there are actually a few putative zinc binders, okay? And so we were really drawn to these two nodes. This node is actually annotated as Yersinia-bactin, which is a known iron-binding molecule. But zinc binding of Yersinia-bactin had never before been shown. And so we were very captivated to see that Yersinia-bactin was likely binding zinc in this context. And we also found that there were various other sort of truncations of Yersinia-bactin that seemed to also be binding zinc. And so from there, we were actually able to do NMR to show that indeed these molecules are binding zinc. And in collaboration, we were actually also able to go back into a mouse model and show that Yersinia-bactin does indeed facilitate E. coli Nissl colonization of the inflamed mouse gut. And so this was an experiment, again, run by Huey and Manuela Raffatello, right? Where essentially they had wild type mice. They induced this dextran sodium sulfate model of colitis, right? So this is essentially a gut inflammation model. And then they orally inoculated with a 1-to-1 mixture of wild type versus knockout E. coli Nissl. And what you can see looking at this somewhat complicated chart is you can actually start to see that the wild type is exhibiting a significant growth advantage, a significant competitive advantage over the mutants that are lacking in zinc transporters and specifically lacking in the ability to either produce or take up that Yersinia bactin molecule. Okay, and so we're quite excited because we used our new method to find that Yersinia bactin, an uncharacterized zinc binder, is indeed binding zinc. And this zinc binding seems to actually be relevant in the context of gut inflammation. Okay, but because I think this audience is really interested in sort of eco-metabolomics, right? I wanted to sort of show another example. And this example is actually starting to investigate iron binding molecules produced in an ocean ecosystem, okay? And so during my postdoc, I was very lucky to be in the Dorstein lab, right? To have access to tons and tons of different samples. And so actually, Daniel, who is also participating in this workshop, Daniel went on a research cruise here on this vessel pictured here and collected a number of samples. And so we thought that it would be really interesting to see like just how far we could actually push this method, right? Because these ocean samples or the dissolved organic matter samples are really some of the most complex samples that we ran in the Dorstein lab, right? And so we wanted to see, could we also find metal binding molecules even in these super, super complex samples? Okay, and so the question was, do we actually observe siderophores in the California current ecosystem samples that Daniel and Professor Lahini Aluwari collected as part of this CCE cruise? Okay, and so the California current ecosystem actually refers to this productive upwelling ecosystem in the North Pacific Ocean that's fueled by a seasonal upwelling of cold and nutrient-rich waters. And as you can see, when this upwelling occurs, it's bringing up nutrients like iron with it, okay? And so samples were then collected by Daniel and others near the point of conception, and this is off the coast of Central California, as you can see here. And so just a bit about how the sampling was done. So it was performed at 26 sites at 6 depth profiles with 2 replicates per site, okay? And so you can see the different cycles here, and we'll actually, for the remainder of our native metabolomics investigation, we'll be focusing on cycle 3. And that is because this sampling actually followed a phytoplankton bloom with cycles 1 and 2 in the upwelled waters at the start of the bloom, and then cycles 3 and 4 in the waters after the bloom. Okay, and so we hypothesized that looking at cycle 3 might give us the best opportunity to find siderophores, as after the bloom, the waters would be most nutrient deficient, right? Which would mean that organisms would sort of need to produce siderophores to actually acquire that limited amount of iron. Okay, and so as I told you, these CCE samples contain unprecedented chemical complexity, right? And you can see a big molecular network here. This is sort of way more complex than that small molecular network I showed you previously. And so this contains more than 5,000 nodes, and at the time, a very low annotation rate of only 4%. So a lot of this is what we can call chemical dark matter. Okay, and so we were specifically interested in looking at samples from cycle 3, right? As I mentioned, the hypothesis was that nutrient depletion during the phytoplankton bloom may result in siderophore production. And so again, we ran these samples using our post-LC infusion method, and we were quite intrigued to find that indeed it looked like we were finding a number of siderophores, right? And so one of these, interestingly, is the molecule of domoic acid that I've been showing as an example molecule throughout both talks, right? And so we found domoic acid, and we also found the domoic acid iron binding dimer, okay? And so I presume many of you in the audience have heard of domoic acid, but if you haven't, this molecule is actually quite famous because it's the neurotoxin that made these birds kind of go crazy in the famous movie of Alfred Hitchcock's The Birds, right? But really, domoic acid is quite an important molecule, and specifically, it's even more important to start understanding the fluxes of these molecules as the climate changes and oceans warm, right? And specifically understanding how domoic acid may interact with metals can provide additional knowledge into really what is going on with this molecule, okay? But in addition to domoic acid and finding that domoic acid iron binding occurring, we also were able to find a number of unannotated and putatively novel siderophores, right? And so we haven't yet followed up on what these molecules are, but an interesting thing to do would be to correlate our metabolomics with the genomic sequencing results that we have to try to find the producer organisms of these putative iron binders, and then actually culture those organisms and look for those iron binding molecules in culture. And so I wanted to bring up this sort of eco-metabolomics example because I think that perhaps looking for metal binding molecules in these environmental or ecological samples could be of interest to some of you guys. Okay, but with that, then sort of the next question became, like, could we extend this search even farther, right? Because I've told you two stories. I've showed you that we've been able to identify some new metal binding molecules, right? But the question then that I had was like, well, we have tons of public data in the GNPS Massive repository, and you'll continue learning about this in the coming days of the workshop. And so my question was like, could we actually develop a way to mine that public data to sort of just find all metal binding molecules that might exist, right? And to understand, like, what contexts do we see these metal binding molecules in? Right. And I was hoping we could take advantage of these public repositories, right? And so what Ming, who was also a postdoc in the Dorstein lab and now is a professor at UC Riverside, and I did is we thought we could actually take advantage of this new tool that Ming developed called the mass query language to actually start using it to find metal binding molecules broadly. Okay, and so Ming will actually be talking about MassQL on Thursday here. So you'll learn a lot more about this, but just quickly, I'll tell you a bit about how we can apply MassQL because it's understandable, it's flexible, it's scalable, it's reusable. It seemed like a perfect tool for us, right? And so what you can do using MassQL is you can really query for any mass spectrometry pattern in data that you have, right? And so here's just a super simple example. Essentially, we can query MS2 data, right? And so here I'm going to be showing you MassQL description and then a description in English. We can query for an MS2 product, for example, with a given tolerance. We can query for other peaks, etc. And so what I thought we could do is we could actually try to write a query for metal characteristic isotopic signatures, right? And if you remember back to the sort of data acquisition talk, I mentioned how metals often have very unique isotopic signatures. And so we can just look at iron, for example. Now iron, the most abundant peak exists as iron 56. With some number of C12, right? But then iron exists naturally as both iron 56 and as iron 54, right? So for every 100 atoms of iron 56, we have 6.3 atoms of iron 54. And so because of that, iron in the mass spectrometer looks like this, right, where we have this diagnostic iron 54 peak, right? And so we would not have this peak if we only had carbons. This peak is diagnostic of iron, right? And so what I thought we could do is I thought we could actually write up a query that would specifically look for this iron specific fingerprint, right? And so that's what I have written here, right? So this specifically looks for this iron 54 peak in the MS1, right? Relative to this, this peak X. And we're looking with a specified abundance, right? Then we say we also need the presence of a base peak. We also need the presence of this iron 56 C13 peak. And additionally, we also want to find both iron-bound and proton-bound peaks for additional confidence. And then we want to find the MS2 associated with that so we can do molecular networking. Okay, and then we wanted to further optimize our query for iron. And so what we did is we looked at this siderophore database, right, which is a database containing hundreds of siderophores. And we actually took a computational approach, right? So we actually computationally predicted each isotope pattern of every single siderophore in this database, and we plotted those, right? So here we're looking at M over Z versus relative abundance. And what you can see is we can actually look at each of those peaks, right? So here is that iron 54 peak that I showed on the previous slide. And we see that always has an abundance of 6.3% relative to that base peak, which always has an abundance of 100%. But we can refine our query by looking at these other peaks, right? So the X+1 peak, for example, depends on molecular size, right? Because increasing the number of carbons means that we will be increasing the abundance of this peak, right? And so what we could do then is we could actually fit lines to generate our query, to develop a more complex and more robust query. Then we tested our workflow on 2 data sets that we'd run previously. So actually that E. coli Nissl data set, along with a data set of fungal siderophores. And then we could actually run our query on all of GNPS and Massive. And so that's what we did. We actually searched all 300,000, even more actually, files, and 681 million unique MSMS. And we were able to actually find all putative iron binding molecules in public mass spec data, which was really exciting. From there, then, we were able to actually highlight annotated iron binding molecules by coloring in the nodes in orange, while the remaining gray shaded nodes are molecules without an annotation, okay? And so we found some known siderophores, right, such as Yersinia-bactin, which is the molecule that I described previously, that now we know can bind both iron and zinc. But the thing that's really captivating to me is the fact that most of this chemical space remains unknown. And so it suggests actually that we're sort of underestimating the amount of chemical space that does interact with metals. And so now the goal is to try to elucidate what are all of these iron binding molecules. Okay, but the other question that really intrigues me is actually, can we extend our search to sort of metals beyond iron and zinc, right? So iron and zinc are sort of some of the most well characterized of these redox active metals, right, that we know are important in biology. Those are the ones that are shown in green. And I'm really interested in understanding, can we find metallophores for other non-iron and non-zinc metals? And so I'll just close by telling you a quick story of how we've actually started like broadening our thinking about what metals are relevant and what metals are actually needed in biology. And so I'll tell a quick story about actually the lanthanide metals. So it was only about a decade ago that these lanthanides were actually found to play a role in biology. And this was specifically in methylobacteria, or bacteria that live in the phyllosphere on plant leaves, right? They actually use lanthanides to perform methanol, well, in a methanol dehydrogenase, right, to metabolize methanol that's produced by plants. Okay, and so this work was done in collaboration with Professor Ceci Martinez and the grad student in her lab, Alexa, along with Professor Lena Dauman, and also actually Daniel Petrus, again, who you'll be hearing from soon. And so these two really kicked off this work. And what they found was this organism, Methylobrum extorquens AM1, actually responds to insoluble neodymium. This is a lanthanide, right? By sort of changing its gene expression, right? And so they found that a number of genes are actually upregulated when there's lanthanide limitation. So basically, when these organisms exist in the presence of insoluble lanthanides, meaning they can't actually get at it, versus in the presence of soluble neodymium, right? And so we see a change in a number of genes. And so what are these genes? Well, you can see here, interestingly, that when we look at the BLAST-P to the next related protein, if you're used to looking at siderophores, you can see that actually a number of these proteins are, well, either siderophore synthase components or machinery involved in making siderophores. And so that was quite interesting. And it suggested that maybe these genes are actually important in making a molecule that's secreted to take up the lanthanide. So not a siderophore, but we can sort of call it maybe a lanthanophore, right? And so what we did is we wanted to figure out, are these organisms making some secreted molecule that can actually bind to lanthanides? And so what Alexa, the graduate student in Ceci Martinez's lab at Berkeley, did is she made the genetic mutant, right, where she knocked out that cluster of genes, right, which we can call like a lanthanide chelation cluster. So she knocked that out. In addition, she made another mutant in which she overexpressed that biosynthetic gene cluster. And so we ran the mass spec data, and this was actually just run using standard differential abundance, MSMS. And so you can see that mass spec, again, is very powerful when we have access to, for example, wild type versus a knockout and an overexpression organism, okay? And so we hypothesized that if a molecule were being produced, we'd see it only in the case of overexpression, which is shown in blue, and wild type, grown in different metal levels. Wild type is shown in orange and red, but we would not see this produced in the knockout, okay? And so we were quite excited because when we actually ran these results, this here is run in negative mode, we found that only one molecular family was actually present in overexpression and wild type, but not in knockout, okay? And that was this family here. Now, when we made a volcano plot, we found that indeed one of those molecules, this feature 797.4, was the most increased in wild type as compared to knockout. And you can see that quantified here. So again, very increased in overexpression and in wild type as compared to knockout. And when we looked a little more closely at the MS2 spectrum associated with that molecule, we were very excited to see that indeed it actually looks somewhat like a known siderophore, the siderophore rhodopetrobactin. Right now, rhodopetrobactin, instead of having the hydroxyl just at this 4 position, it actually has another hydroxyl group as well. But we were able to annotate this molecule based on the structural similarity to rhodopetrobactin. And we were able to find that perhaps this is the first biological small molecule lanthanide chelator. Okay, and now we're very interested to start to understand whether this is specific for lanthanide versus iron and to better understand sort of the metal binding properties of that. And that's something that our collaborators are now working on. And so with that, I'm going to close my talk. So I hope that you can see that we've started developing some new experimental and computational approaches for discovering metallophores and metal binding molecules from biological systems. And actually, really excitingly, Daniel and I have taken similar approaches to also look for protein ligand binding, and Daniel has really continued on in that direction to have some really exciting findings. And so maybe he'll tell you a bit about that if you ask him during his seminars. And additionally, we've started applying these tools to systems of increasing complexity to find a new zincophore, a new lanthanophore, and to start fully elucidating the iron binding metabolome. And so with that, I'd like to thank a number of people. So first off, my lab now at DU, my lab has grown, so here it is, along with a number of mentors and collaborators during my postdoc, specifically my postdoc mentor, Peter Dorestein, and various members of the lab that really helped out. Additionally, I'd like to thank all of my collaborators, and I'm just highlighting Ming and Daniel, who I also highlighted within the talk, but they'll both also be presenting at this workshop as well. And so definitely have some time talking to them. They're both really amazing scientists and were very instrumental in this work. Additionally, these other collaborators, so Manuela Raffatellu with the Zinc story, Ceci Martina and Alexa with the Lanthanide story, and then my collaborators, Emily and Dr. Zach Reitz, who also helped in a number of these studies and has been very essential as well. And so, yeah, with that, I'm happy to take any questions. And yeah, thank you again for listening to my talk.
Thank you very much, Allegra, for such a nice talk. We have already one question here.
Okay, awesome.
Hi, yeah, thank you for the question. Thank you for the great presentation. I had actually just like a couple of weeks ago read the native metabolomics paper you just mentioned from Petrus on the proteins, and it's just I went down the rabbit hole since then, so it's really fortunate to be able to talk to you now.
Awesome. Nice.
But so one of the questions that had come up when we were going over these papers in our research group was the technical limitations of it. So in regards to like what LC-MS/MS setups actually allow to do these native metabolomics experiments, because I saw that for the protein one, I think it was done on an Orbitrap, whereas we primarily are working with a QTOF in our lab, and whether or not that would still be possible, maybe not for proteins, but maybe for like these metal chelating experiments like you were saying. And then furthermore, if you do set this up, how easy is this to integrate into like your standard metabolomics workflow? Does it need to be like one or the other? Is it easy to switch on and off, basically?
Yeah, no, so those are great questions because it is exciting to know that people are interested in using these workflows too. So as you did note, the Petrus paper and then our original paper, both of them were done on the Q-Executive Orbitrap. That is what I have in my own lab as well. I don't see any reason why we shouldn't, or why you shouldn't be able to modify the QTOF to do this experiment. We had always spoken about trying it on a QTOF to like make this more generalizable, but then we just never just didn't get around to doing it. But I think it should definitely be doable. So now, actually, Daniel and I both have sort of integrated LC systems where we have two pumps. So essentially, we have one pump pumping our water acetonitrile gradient, and then we have one pump pumping ammonium acetate. to raise the pH. But when we first started playing around with this, what we did was we actually took like an old HPLC pump that was not integrated, and we just teed that in. And so we controlled that gradient from a different computer. And so I think if you didn't have just, you know, like an integrated, I assume you don't have an integrated pump already, you can always find, well, maybe you can find sort of an HPLC pump that's lying around, not getting used, and just tee that in, because for most of these experiments, I'll just use sort of a consistent ammonium acetate infusion throughout. So it wouldn't be a gradient, it would just be a consistent flow. And then with regards to the infusion, we use the syringe pump that is also integrated into the Q-Exactive. But again, I don't see why you couldn't just, you know, get a syringe pump and use that for your metal infusion. So basically all of this is to say, I think that the method would be very much amenable to modification for the QTOF. And we have a QTOF that's not in my lab, but it's in the department. And so this might be something that we should also start looking at so that more people can use the method. So that hopefully starts to answer your first question. I'd also be happy to talk more, like if you do want to set this up, like to troubleshoot with you if you want, obviously offline in the future. Your second question, I forgot what your second question was. Can you remind me?
Yeah, so the second question was just how easy is it to, so when you have the system set up, is it easy to switch between workflows?
Yeah, so I guess that's a good question. So yeah, we sort of went with this integrated pump and using the integrated syringe, so the integrated quat pump of our LC and the integrated syringe pump to keep it really easy, because yeah, essentially we'll do sort of a normal experiment and then we'll do a metal experiment. We switch all the time, right? I think if you have like an external HPLC pump and maybe like an external syringe pump, it might be slightly less straightforward, but I think it should be totally doable, right? It just might take slightly more time. But I think the moral of the story is it's pretty easy to switch between. The only thing I would suggest is we do start infusing high levels of iron sometimes. And then I would definitely make sure you're cleaning out your tubing and your source well, or we try to use dedicated tubing sometimes so that we don't see metal addicts all the time, right? Because metals would be rather sticky. So definitely having that cleaning in between is going to be important. Yeah, so hopefully
thank you so much.
Yeah, for sure. Other questions?
Other questions? I have another question regarding this metal infusion.
Sure.
How much noise do you introduce into your data? And how do you remove this noise that you introduce by the metal infusion?
Yeah, so that's actually a really good question, because, and something we've sort of been thinking about for a while now. So we do introduce noise into our data because, well, our metal salt that we infuse, it's the highest, like we've bought sort of the highest purity that we can, but yeah, we're seeing there's lots of signals in our salt, right? So our background level is quite high. So we do see more noise. Usually what we've done is we'll always do a blank with metal infusion, and then we use that for blank subtraction. So subtracting features that are about that are present in the blank, also in our samples. to try to get rid of noise coming from the metal salt and perhaps the ammonium acetate. So that's the first strategy. The second strategy is we typically will run our samples with and without infusion. We'll run them also at low pH and at neutral pH to see sort of if we have lost any features, like when we've sort of changed our setup. Usually our features of interest, which would be the metal binding features, would not be lost in the noise, at least in our experience so far. So does that answer your question, sort of?
Yeah, thank you very much.
But that's something that we're continuing to try to optimize, I guess, because it is an issue. The other, I guess the one other thing we've done is essentially creating like an exclusion list. So we would run our blank with metal infusion, see which MS2s we're getting in that blank, and then we would put all of those in the exclusion list, right? So that we're sort of removing, well, so that we're not triggering all of the MS2s just from contaminants in our metal interacting with the blank. So that's another attempt to reduce the noise. But yeah, that is certainly a challenge that we're still working on.
Okay, perfect. So thank you very much. Somebody
Yeah.
has another question? Yes.
I was wondering, so you set up this specialized method to find molecules that bind iron. So how come you found so many in the public databases? Because they didn't use specialized methods at all, did they?
Yeah. So that's a really good, that's quite a good question. Yeah. So what we looked at, so when we were thinking about sort of what fraction of molecules would be binding metals versus what wouldn't, we found that it seems like about 30-ish% we would of metal binding molecules, we would still see the metal adduct. So it's not like we never see the metal adduct, it's just that we often don't see the metal adduct, right? And so I think we still find some of these metal bound species in the public data, but I do think one of the limitations with that approach is that we are missing many metal bound species, and these would often be like weaker metal binding or molecules that are less abundant. We probably wouldn't be seeing those metal adducts, but yeah, that's a good question and sort of a good thing to clarify. It's not that we never see the metal adduct, it's just we see it less frequently than we see the proton adduct in many cases, especially in the case of weaker binders. Does that sort of answer the question?
Thank you very much.
But that's
More
a great
questions?
question. Yeah.
More questions? No? Okay. So please help me again to thank Allegra for such a nice talk, for introducing all the data acquisition possibilities and for showing us the great work that she does in the USA. Thank you, Allegra, again for participating in this workshop. I hope to meet you in person at some point soon. And
Yeah.
I have the pleasure to invite you next time for the next round of these workshops here in Germany.
Yeah, so thank you so much for the invite. And I'm sorry I couldn't be there in person. It's always more fun to do an in-person talk, but I'm glad I got to participate. So I hope everyone enjoyed it. And I hope you have a great rest of the workshop.
You're already included in the list for the next one.
Oh, thank you.
Okay. Thank
Great.
you very much. I think we have to close the session today, Allegra, here, because these participants must be very tired of the
Yes.
a lot of information that they gathered today.
Yes.
Thank you very much again, and hope to see you soon again.
Thank you. Awesome. All right. Bye-bye.